Today we're going to talk about the central dogma of biology. And so first off, look here where the red writing is, and it says DNA goes to RNA and RNA goes to protein. That is the central dogma. DNA going to RNA, that is the first process, and it is called transcription. RNA going to protein, that is the second part, and that is called translation. So those are the two parts of it that we are going to learn. If we look at this a little more clearly, we can see DNA going to RNA in this top part here. That's the transcription part. And in transcription, with DNA going to RNA, it's going to mRNA, which stands for messenger RNA. Okay, so that is that first place. And it takes place in the nucleus. Okay, so transcription takes place in the nucleus, and it's where DNA is coded into mRNA. So that is the process that we'll be looking at today in more detail. Tomorrow we'll look at the second part of this where RNA goes to protein. That's called translation. So first stop, let's look at the steps of transcription. The first is initiation. Here we have RNAP, RNA polymerase. And it's going to attach itself to the DNA. In this case, the DNA is in black. And the top strand is going to be the coding strand. Okay, so that's going to be the strand that is not used, and the other strand is going to be the template strand. And that's going to be the strand that is actually going to be used to make the new mRNA. Then, the second step is called elongation. Okay, so initiation we have in here. And then we have elongation. Elongation is when nucleotides continue to be added to the mRNA strand. Notice the RNA polymerase continues to go down, and the mRNA is the blue there, and it continues to be elongated. Finally, we have what we call termination, and that's where no longer are there nucleotides added to that. So those are the three steps of transcription. Once transcription has taken place, we have what's called post-transcriptional processing. And so we're going to look at post-transcriptional processing. And post-transcriptional processing is going to be shown right here. Two things happen. One, a 5' end cap is added, and that's that blue dot there. Also, a poly A tail is added to the 3' end of the mRNA. And then that mRNA is going to have one more process happen that is in this post-transcriptional processing. The other thing that's going to be happening here is that we're going to have part of this mRNA that's not actually going to be used for making proteins, and those are called introns. And introns will be cut out of that mRNA. So they're not even going to leave the nucleus. They are going to be cut out of the mRNA, and then finally the exons, which is the part that will be made into a protein, are going to come together, and they are going to leave the nucleus through these nuclear pores. And there in our initial picture, we can see the mRNA leaving through a nuclear pore. So this is the process of transcription, and then tomorrow we'll be going over the process of translation.